Okay, during our last lecture, we started discussing protein synthesis. First of all, do you recall what protein synthesis means? We are making proteins. Yes, we are making proteins. And where are the instructions to make proteins stored? In the DNA. In the DNA. And where is the DNA located? In the nucleus. And how much of the DNA is used to make a protein? A segment. What's that segment called? A gene. And so what we just started discussing was how we get from DNA to protein. And we discussed copying just the gene, and that being a RNA copy, and that RNA copy traveling out and attaching itself to a ribosome, where it was translated into a protein. But we got very basic. We didn't go into detail. So today we are going to go into detail. I'm going to start with where I stopped in the last lecture to review exactly what's going on during transcription, and then we will talk about details of transcription.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Okay, so once I showed you that, we talked about how the enzyme, which the enzyme shown in that video was RNA polymerase, how did it know which gene to copy? What was there that told it, hey, this is where a gene is located? The promoter, okay? The promoter, and I heard someone say stop, and that's going to get us where the termination part is. But what tells us where to begin? AUG, and AUG is a start codon. It's the only start codon. And do you remember what precedes that start codon to kind of tell that enzyme you're coming up to a gene? It's kind of like a yellow light. I said it was kind of zipping along, that RNA polymerase is zipping along, and as it gets closer, it kind of slows down when it reaches this segment. What is that segment called? It's repeating A's and T's. The TATA box, or the TATA box. The TATA box comes right before it, then we have our start codon. In transcription, so here I have the big picture. The information from the DNA is transferred or copied into what we call a complementary strand of RNA, specifically messenger RNA. There are 3 steps to transcription: initiation, elongation, and termination. just based on the conversation we had, you already know what occurs during initiation. And what does initiation even make you think? This is where it's starting. And the only reason we know where to start is because what has to be present? An AUG, which is a start codon. And remember, at the beginning of that video, it was kind of hanging out there waiting for it to be verified. So we're at that start codon. Right before that, or in that region, is what we call the promoter that tells us, hey, you found the correct gene. Now we just need it to be verified. So the RNA polymerase enzyme, once initiation has taken place, begins the process of elongation. What does elongation even sound like logically? Something's elongating. It's just making it bigger. During this step, so we've started. Now during elongation, we're just adding one nucleotide at a time. So adding a letter and 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 a letter, and we're going to continue to do that until we hit a termination or a stop codon. What are your three options for stop? UAA, UAG, UGA. Those are your options for stop. As we're going through transcription, RNA polymerase is doing the work. Are we copying both strands of genetic material? No, just one. Quick question and throwback. What process copies both strands? DNA replication. DNA replication, which occurs during interphase, specifically S phase, right before mitosis. I heard someone say mitosis, and you're not wrong for that, but just knowing you need to make sure you can distinguish between DNA replication and protein synthesis. Okay? Be able to distinguish those. And I had that slide during our last lecture that indicated the major differences. So initiation is where it begins. The RNA polymerase will bind at the start codon, and elongation is as it's adding each nucleotide. And again, it is RNA polymerase functioning during protein synthesis. Termination occurs when it hits a stop codon, and then, of course, that messenger RNA is released. Now, here I've added two things. After termination, if you're a prokaryote, you are all done. Because if you're a prokaryote, how many sets of genetic material have you inherited? One. Because you had one parent, you were produced by the process of binary fission. However, if you are a eukaryote, you copied or you transcribed both expressed and non-expressed segments of your gene. So we need to go back and get rid of what we don't need. So if you are a eukaryote, there's an extra set of steps that has to occur. So of course, this applies to you and I. Bacteria would be done at this point, but eukaryotes, they have a little bit more to do. So transcription is transcribing or copying the DNA, which is located in the nucleus, because it cannot leave, into a copy which we call messenger RNA. If you're a prokaryote, you're done. If you're a eukaryote, we go through what we call messenger RNA processing. During messenger RNA processing, the very first thing that occurs, so here's our DNA, and once it is transcribed, I just want that segment, that gene. The very first thing that occurs is we put a 5' cap. There is a guanine cap, so a G cap put on the 5' end. When I initially learned this, it was kind of confusing to me, and so I needed to figure out how does this apply to what I understand. Maybe this will help you. I call it now, this is my 5' captain, because this cap here will guide this messenger RNA to the correct ribosome. So it's responsible for making sure that if this is the messenger RNA for an I protein, that it makes it to an I ribosome. It's responsible for guiding it to the correct destination. So after it is transcribed, and this is in eukaryotes only, a 5' cap is added. The second step, a poly A tail. What does poly mean again? Many. And then A is talking about the nucleotide adenine, yes, or adenine. We're going to put multiple A's at the very end. And by multiples, I mean like a couple hundred or so. Just a poly A tail. The purpose of this tail is twofold. The purpose of this tail is twofold. The first is to provide this short segment of genetic material with stability as it's moving throughout the cytoplasm. First function is stability, molecular stability. The second is longevity. It allows that messenger RNA to be translated multiple times, not just one time, but multiple times. So again, the purpose of the poly A tail is twofold. Stability, molecular stability, and then molecular longevity. So that messenger RNA strand, once it's processed, will be used to translate multiple proteins, not just one. Will be used to translate into multiple proteins. It itself will not translate proteins. I apologize. So we've transcribed it, we cap it, we put a poly A tail. Those are the first two steps. Next, notice here how it says exon, intron, exon, intron, exon, intron, and so on. They're repeating. Exons are the portion of your gene that are expressed. I memorized exons expressed. The introns are the non-expressed aspects of that gene. They are the non-expressed aspects. So when I go to translate it into a protein, guess what I don't need? The introns, the stuff that's not expressed. In fact, your book refers to it as junk mail, something that you have but you don't really use. Something that you have but you don't really use. So during this third part, we are going to splice out introns. Splicing introns. It's called splicing, or messenger RNA splicing. All that means is that there is an enzyme, and you don't have to know the name of this, but the enzyme's name is spliceosome, so it kind of works out. But the spliceosome will get on top of that intron, cut it here, and then seal that back together, seal the exons together. Cut here, seal it back together. So the introns are spliced out. At the end of this process, you should have nothing but exons with a cap and a tail. At the end of this process, you should have nothing but exons with a cap and a tail. So for example, if this was a transcribed gene, full gene, for my eye color, I told you that I have blue eyes. You can tell I have blue eyes. So all of these segments here are blue. Are these exons or introns? They're exons, which means that they are expressed. So all of these blue segments are exons. I told you, though, that although my mom has blue eyes, my dad has brown. So all of these darker lines are going to represent the color brown. But because you can tell by looking at me I do not express these brown ones, what are these referred to as? Introns. So during this process of splicing, what occurs is an enzyme, again called a spliceosome, comes in and it cuts out each intron. Each intron is removed, and then all the exons are sealed back together so that all I have is the genetic material that is expressed. Of course, along with this would be my 5' cap and my poly A tail. At this point, eukaryotic messenger RNA is completely processed, and it's what we refer to as mature messenger RNA. This only occurs in eukaryotes. Only occurs in eukaryotes. Questions so far? Okay. So, transcription, one more time, I'm going to show you the video from beginning to end. The goal is that you understand exactly what's going on, what's taking place, why it initiated in a certain position, and why it terminated in a certain position. So, a final summary for transcription.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go.
RNA polymerase.
The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Why did it stop again? It hit a terminal codon or a stop codon. And the combinations of the options for that are UAA, UAG, or UGA. So after transcription, that messenger RNA rolls out of the nucleus after it is processed, and don't forget we have to cap it, tail it, and splice it. And it heads itself to a ribosome. So this is what the aspect step 2 that we're going to study now, translation. Overview to begin with.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein.
Your protein is up there out at the top.
Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules.
So here is a transfer, amino acid, anticodon would be down here.
Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process.
Notice that there are 3 stopping points.
The RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome.
And the protein is folding because the shape matters.
Any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
And why did it let that protein go? It was done. It will hit what we call in just a moment a release factor. It will hit a release factor. So we're going to start this process, translation. And I want to begin with the transfer RNA. Because to this point we've said messenger RNA, messenger RNA, messenger RNA. And if you did the lab last week, then the terminology I'm about to use should just be a refresher for you. But transfer RNA has 3 components. Transfer RNA has 3 components. It has the actual transfer molecule, it has an amino acid, and it has an anticodon. What I have pictured here is straight out of your text. It gives you 3 different depictions of the exact same molecule. So the one you just saw in the video followed this shape. The one that you will see in the notes on the PowerPoint after this slide followed this shape. But they are all the same thing, and I will point out what they represent. Right here is the transfer molecule, the amino acid, the anticodon. There's 3 parts. For this one, here is the transfer molecule, the amino acid, the anticodon, all 3 parts. Right here is the transfer molecule, the amino acid, and the anticodon, all 3 parts. These are just 3 different ways that you will see this molecule represented in your text. Okay? So I wanted to make sure to explain that they are the same thing. It just depends on what model they're using. I also use the term anticodon each time I explain the transfer RNA. The anticodon cannot just be some random sequence. The anticodon has to be complementary to the codon. The anticodon has to be complementary to the codon. So, for example, if my messenger RNA codon was AUG, my messenger RNA codon was AUG, the anticodon would have to be the complement to this. What's the complement to A? If it were DNA, it would be T. But am I in the nucleus anymore? I'm in the ribosome. So the opposite of A is not T, instead it is U. The complement to U is A, fantastic. The complement to G is C, fantastic. This is the anticodon for the transfer RNA. So what I mean is they have to be complementary. questions there? Okay. Now, when we look at the anticodons, you need to keep in mind that, do you remember that our genetic code is redundant or degenerate? I had mentioned this last time. What does that mean, that our genetic code is redundant or degenerate? Yes, ma'am, that is exactly correct. I can have multiple codons, and I put this chart back here so we could reference it. Multiple codons code for the same amino acid. Because remember, how many total amino acids do we have? 20. But we have 61 actual codons that we can use for amino acids. Technically it's 64, but these three do not have an amino acid. So we subtract those three and have 61 codons that can actually code for an amino acid. So what the wobble hypothesis says is that when we're translating, the transfer RNA really just needs to get the first two nucleotides right. And let me show you what I mean. As long as they get AC correct, AC, 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 does it matter if it comes out ACU, ACC, ACA, or ACG? No, I'm going to get the same amino acid. So what the wobble hypothesis says is, as long as I can get the first two nucleotides correct, that last one, there's a little bit of flexibility. Now, is that always the case? The answer is no, it's not always the case. Like, it wouldn't work that way for tryptophan. But like for proline, alanine, valine, threonine, all those, it works out well for. Okay? So the wobble hypothesis just says that as long as we can get those two right, the first two nucleotides correct on that transfer RNA, that anticodon, the third one, we have a little bit of flexibility. And this is only because our genetic code is redundant or degenerate. Okay? And we used those adjectives last week to describe it. Questions here? What's the matter, Andrea?
I'm just comparing the number of amino acids.
Uh-huh.
There's some that are repeated, right?
Yes.
Okay.
Yes, ma'am.
Sorry.
No, you're okay. You're okay. I just didn't want to keep going. Look, like serine is here and here.
Okay, good.
Yeah? Of course. Then there's the ribosome, which is the location of translation. I would love to just say, and that's the ribosome. But there's the anatomy of the ribosome that we have to understand in order to get the process in its entirety. So a ribosome has two parts. It has what we call a small unit or a small subunit and a large subunit. When this ribosome is not working, these two are not together. And I'm going to go back to the video in just a moment to show you that it's not just sitting there waiting for something to happen. These two pieces are normally separate if they are not working. The 5' cap on that messenger RNA will guide that messenger RNA to the small subunit. The small subunit will say, oh, this is correct. And in the event that it has been verified, then the large subunit will slam down. I'm going to show you what I mean, and then I'll come right back to this, because it was depicted in that video. So it's
all the components of a molecular machine locked together around the
small
RNA
subunit,
to form a
verified, large subunit. So the large subunit and small subunit are not together just hanging out waiting. The small subunit will verify that we have indeed got the messenger RNA that we can translate, and if it is verified, then the large subunit will slam down. Okay? This prohibits or prevents the synthesis of the wrong protein, because what if when it got to this point, it said, wait, you're the messenger RNA for an eye, but I'm this small subunit for bone. I don't know how to read you. If that occurs, is that large subunit going to slam down? No, it won't. It can't do it. It won't do it. So, when that large subunit slams down, it immediately begins to translate. Now, whenever we were showing the video, I said, watch, there's 3 spots. There's 3 spots. And I wanted you to just get that in your head so I could explain what each of these spots is used for. I call them bays because it makes me think of like a big garage door, so like a bay. But they go in this order, A, P, E. So it spells ape if you follow them in the correct order. In the A site, the amino acid is verified. Is this the right amino acid? If it is correct, it jumps over to the P site where a peptide bond is formed. Why does it have to be a peptide bond? I'm making proteins. And when I join amino acid to amino acid, I dehydrate and I create a peptide bond. That happens at the P site. And then once the peptide bond is formed, that transfer molecule shifts over to the E site, and E is for exit. And it releases it. So again, at the A site, the amino acid is verified. So the transfer molecule brings it in, the anticodon and the codon fit together. It's been verified that we are indeed the correct one. So we shift over to P. That transfer molecule will allow for a peptide bond to be formed between the amino acids. Then once that amino acid is gone, the transfer molecule hits the E site or exit, and it's released. It'll go back and pick up another amino acid, and it'll come back when it's needed again. So the transfer molecule will go from A, P, to E, and it then will leave. I put here what each site represents. So A is where the amino acid is verified. P, a peptide bond is formed. And E is where that transfer molecule will exit the ribosome. Here, during translation, big picture, information that's coded in the messenger RNA is now translated into an amino acid sequence. What's an amino acid sequence? A protein. What's a polypeptide? Many peptides, so what am I making? A protein. Don't forget, 9 times out of 10, it's not just going to say, hey, we're making proteins. It may say, we've created an amino acid sequence, and you need to know that that is a protein. Or that we're constructing a polypeptide, and you need to know that that is also a protein. Okay? So just knowing the words is more than half the battle. It's like 3/4 of the battle. If you know the terminology, you're more than likely going to answer it correctly. Okay? So, right here it shows just the small subunit. The messenger RNA will attach. It will be verified in the event that the messenger RNA has indeed found the correct small subunit. The large subunit will slam down. And it will then begin to translate. There are 3 steps to translation. Do these look familiar? Why do they look familiar? They're the exact same as transcription. Initiation, which means I'm doing what? Starting. Elongation. I'm adding, elongating that protein or amino acid sequence or polypeptide. And then termination. I'm done. I'm done. So we have the same 3 steps: initiation, elongation, and termination. And I enjoy that because it's easier to study. If you know we start, we continue to add until we hit the stop. Now, I kind of want you to see in I'm going to point a couple things out. But this process happening like with in a step art form. So here my messenger RNA has been verified by my small subunit. The large subunit slams down. I really enjoy this example because it uses actual codons. Like this is the actual start codon, AUG. And the actual start anticodon is UAC, the complement to that. And it's attached to the correct amino acid, which is methionine. So it's there, it's at the peptidyl site, which means it's ready to add a peptide bond. Then the next transfer molecule will show up. You can see that it's seated here in the A bay. The A bay verifies that this is the correct amino acid to be added. If it is the correct amino acid to be added, it will shift over to the P site and it will add it to the previous amino acid. What type of bond is formed there? Peptide bond. Notice how the very first transfer molecule has shifted to the E site. What's it going to do once it hits the E site? Exit. And it will continue this process where the next transfer molecule will come in, become verified, add that amino acid to the polypeptide chain or amino acid sequence or protein, all three the same thing. And then they'll hit the E bay, the bay labeled E, which is our exit bay. And it will leave. This will go out and find another amino acid to bring back. So I like this example. I think it shows it running really smoothly. Does anyone have questions on the A, P, or E sites? Okay. I put this slide here because I said it out loud, but it's not anywhere in writing. So during elongation, the transfer molecule will enter the A site where it is verified. And once it is verified, it will then move to the P site, then to the E, where it will exit. So here is elongation in writing versus me just telling you. Of course, after elongation, the next step is termination. During termination, and you can see that we've added amino acid after amino acid after amino acid. So we have a large amino acid sequence or a large polypeptide. Then we have the stop codon, which is either UAA, UAG, or UGA, show up at the A site or in the A bay. As a result, there is no amino acid that comes because stop does not code for an amino acid. It says, hey, it's time to stop, so a release factor, and this says RF, this is straight out of your text, RF, the release factor shows up. I bet you can't guess what a release factor is responsible for. Releasing it. It says, hey, we're done, and then it will release that polypeptide, amino acid sequence, or protein, all three the same thing, and then guess what happens to my ribosome? Breaks apart. Remember, it's not just hanging out waiting for work, okay? It only works when someone asks it to do something. It's not actively looking to be employed. Questions on termination? So here's translation from beginning to end.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form
Small,
a picture factory
large.
called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer
Amino
molecules
acid,
for each of the 20
anticodon.
amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process.
You can see all 3 bays.
Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
Questions on translation? Yes, ma'am.
So when it says it is happening in real time, is it because whenever this small synthetase attaches to the mRNA,
Yes, ma'am.
it attaches that fast?
Yes.
That quick?
Mm-hmm. It either knows you belong or you don't. Yes, ma'am. This process happens very, very quickly. And then, of course, it's showing you this, and you're seeing it happening in one spot, but you have trillions of cells that are all doing this nonstop. So when it says that you're churning out 100 trillion molecules of hemoglobin per second, first of all, hemoglobin carries oxygen to your tissues. So you have to continue to make that. And that means whichever ones I'm releasing now, there's another batch about to follow up. And then those that I've already made and used that can't be used anymore are being disintegrated just as fast. Because if not, then I would have a buildup of that protein. So this does happen fast, but your body is created to be able to metabolize. So build up, anabolic, and break down at the same time in order to maintain homeostasis. Fantastic. Melanie, did you have a question? You just let, okay, no. Okay, just. Okay. Polysomes. Poly, you already know, means what? Many. So a polysome is where we have many ribosomes. Multiple ribosomes are capable of translating the same messenger RNA at the same time. Okay? What this is showing is, this is not showing like a stepwise progression. This is saying that this ribosome was initially here, went through and translated it, and as it got a little as it got a little bit ahead, another ribosome attached. So at the same time, we're able to create multiple proteins using the same messenger RNA. And remember that poly A tail allows for the longevity of that molecule. So it can be, we want it to be used over and over again. It took a lot of work to make messenger RNA. So we don't want to just use it once and we're done. We want to use it over and over again. Now, not every ribosome in your body is set up like this. Not every ribosome is set up like this. But this is an option, a polysome. I also want to point out prokaryotic protein synthesis. What do prokaryotes not have that eukaryotes do have? Prokaryotes are lacking a nucleus. And remember, protein synthesis for us, the first step has to occur in the nucleus, because that's where our DNA is, right? And then it has to be carried outside of the nucleus, that RNA does, to the ribosome where it can be translated. But for a prokaryote, we don't have that problem, because a prokaryote's DNA isn't held in by a nucleus. So what prokaryotes have the ability to do is transcribe and translate at the same time, because they don't have to travel to get to their destination. So we say, and I just wanted to make sure that you understood that concept, prokaryotes have the ability to simultaneously transcribe and translate their genes into proteins. What's the only thing on Earth that's prokaryotic? A bacteria. Fantastic. I taught you this concept in chapter 4, but now that we've made all these proteins, what do we do with them? We have to send them off somewhere. Now, in chapter 4, I told you, and you already know that ribosomes make the proteins, but we have two groups of ribosomes. We have fixed ribosomes, and we had free ribosomes. And those ribosomes that were free throughout the cytoplasm, when they generated proteins, they were to serve the cell. They stayed within the cell. However, the fixed ribosomes would generate or manufacture proteins that would be used outside of the cell. My first question is, what structure were those fixed ribosomes attached to? The endoplasmic reticulum. Which endoplasmic reticulum? The rough. Fantastic. So we studied that the rough endoplasmic reticulum has the fixed ribosomes, and so that all the proteins that were manufactured by those fixed ribosomes were going to be sent out for export. But before they could leave the cell, where were they modified, sorted, and packaged? The Golgi. Yes. Yes. And I love this diagram. This is not from your text. I chose this one because it shows the proteins in different colors. Because I told you that when they get dropped off at the post office, all of them are just dumped off. Like, here's all the proteins we made. And then the people at the post office have to say, hey, wait, these are all bone proteins, and these are all eye proteins, and these are all blood proteins, and so on. So it modifies them, sorts them, and then it packages them. And what do they send those proteins out in? A vesicle. Fantastic. And that vesicle, and these are color-coded so you can see that they've been sorted into the right group. That vesicle will carry those proteins to the cell membrane, but the vesicle won't leave, remember? It stays, it fuses with that cell membrane. What's this process called? Where we push things out of the cell? Exocytosis. Yes. Yes, exocytosis. The process of releasing things from the cell. This was my way of kind of lacing back, throwback information I've already taught you into the concept of how those proteins are processed once they're finished. Fantastic. Now, last topic, but it is a pretty in-depth topic, is mutations. Now, I am going to teach you, or we're going to discuss mutations today, and we're also going to discuss them in our next lecture. But the mutations that we're discussing today are specifically gene mutations, because you and I have been talking about just individual genes. And one gene codes for one protein, right? So if there's a mutation in one gene, it affects that one protein, right? So one gene, it's just a segment of DNA. Now, the second type of mutations that we'll end up talking about, and I want to address it so that I don't leave you hanging, but it's chromosomal mutation. So a whole chromosome, which could have anywhere from 25 to 150, 200 genes on it. So if that chromosome mutates, it impacts more genes. Does that make sense? So when you're looking at the material and the test questions and you're studying, am I dealing with a specific gene? Just one gene that's going to manufacture one protein? Or am I talking about a whole chromosome that could potentially impact a lot of genes, which would be a big deal? Okay? So today we've been talking about genes and then being transcribed, then translated into proteins. So I am just going to cover gene mutations. Gene mutations. And the term mutate means to do what? It's just a fancy word for change. And back when we first began this semester, I said the only thing in biology that is random, crossing over and mutations. So mutations are just random changes. You cannot plan for them. And then when we talked about meiosis, crossing over, that process is also random. But everything else in biology is specifically planned. There's a process that it's supposed to follow. There are 4 classes of mutations at the gene level. A missense mutation that results from a substitution. Nonsense is also from substitution. Silent from substitution. And frame shift that results because of an insertion or deletion. Now, before I teach you these at the biological level, I'm going to teach you at Jessica's level so that you can understand the impact of each of these mutations. So I'm going to leave this slide up, but I'm going to walk through examples of each type of mutation. And I'm going to begin with my gene. Is this a real gene? Is this a real gene? No, it's not. It's just a sentence using 3-letter words, but why did I use 3-letter words? Yeah, so my genetic language is made up of words that have just 3 letters each, right? So this is why I chose this. And this is truly how I learned it well enough to explain what's happening to you. Okay? So the first type of mutation, this is the gene, and when it is transcribed and translated correctly, I get the out product protein that I want. This is my goal. Now, in a missense mutation, which results because of a substitution, 1 letter along the way, and each letter represents a nucleotide, 1 letter is substituted for another. So like, for example, let me do it in a different color. Right here, this A is replaced by a U. This A is replaced by, it's just substituted. So this codon still codes for the same amino acid. That didn't change. But this codon, it has a different amino acid. This one did not change. This one did not change. This one did not change. And when I read this sentence, the cut ate the rat. It's still a sentence. Cut is still a word, but does it make sense? No. We call this a missense mutation. Am I still going to make the same protein? No. This amino acid is different. Am I still going to make a protein? Yes. It's not the right protein, though. Does that make sense? Now, is this a big deal or a small deal? Okay, let's talk about you whenever you become a practicing physician. And they're like, oh my gosh, doctor, what does that mean? Your answer is going to be just like your doctor is for you. It really depends. It really depends. Because this might be a protein that we really don't use or metabolize in high quantities. Or it may be one that we need a whole lot of. The answer is it depends. It depends not only on which gene it is, but on the individual that it's impacting. Quick example, not of a specific gene mutation, but of the impact and why the individual matters. I could take NyQuil, and when I take NyQuil, I am out. Probably 15 minutes later, I'm out, and you can't wake me up. However, my little sister can drink NyQuil, and she'll stay up for 48 hours. It just makes her excited. Our genes are expressed differently. We came from the same parents. Well, I tell her she's adopted, but technically we came from the same parents. It's our gene expression is different. Our metabolism is different. The way we use things is different. So when the question comes up, what does that mean? Or how is that going to impact me? Doctors all the time say, well, it really just depends. It really just depends. So a missense mutation, a substitution took place. It's still a sentence. I can still kind of figure out what it's saying, but I'm making a different protein. One amino acid has changed. One amino acid has changed. That's missense. The next mutation is a nonsense mutation. When a nonsense mutation occurs, this mutation takes one codon, and it changes it to a stop. So what occurs here? The protein is no longer synthesized. It stops right here. In a nonsense mutation, one codon is changed to a stop codon. This is also because of a substitution, but I didn't use your genetic words. I used my own words. So where I should have initially had 1, 2, 3, 4, 5 amino acids, I now only have 1, 2. Because remember, the stop codons, there's no amino acid attached to them. Did I make the same protein? No. And the sentence doesn't make sense. So it's the cat. The cat what? So it's a nonsense mutation. A nonsense mutation results from a substitution that creates a stop codon. So that protein is prematurely truncated. We stop it early. That's a nonsense mutation. The next mutation is a silent mutation. And I want to reiterate that the only reason we have silent mutations is because our genetic code is redundant or degenerate. Okay, but in a silent mutation, again, it's a substitution. One letter is substituted by another, but the sentence still reads the same. So did anything happen to me? The original sequence was the cat ate the rat, and my mutated sequence is the cat ate the rat. This is a silent mutation. I'm still going to make the same protein. I'm still going to have the same outcome that I had initially. So there's no evidence that this mutation has occurred. This is other than the genetic code switching. But this is what we call a silent mutation. A silent mutation, again, a result of a substitution. The fourth and final type of gene mutation is called a frame shift mutation. A frame shift mutation is the result of an insertion of a nucleotide or a deletion of a nucleotide. In this instance, let's say that I insert an A after this C. So, my first amino acid stays the same. My second one, I have my C, but now I have my inserted A. Then I have this A, but what has to happen to this T? It has to go to the next word because all of my words can only be 3 letters. So this one has to shift down. So T-A-T. Now this E also has to shift down. This E has to shift down. And this T is now on its own. Does this make the same protein? No. Does it still make a protein? Yes. It's just not the right protein. The same impact. So we inserted it, and all it did was take everything and shift it down one spot. What do you think happens if I delete? It's going to shift back a space. So let's say I delete this A. So again, my first codon in amino acid sequence stays the same. But my second one, the T shifts down. Now this A has to shift down. So then I have T, E. This T has to shift down. And then, am I still going to make a protein? Probably. Is it going to be the right one? No. I wanted to demonstrate those using a sentence so you could kind of see how it fits together. Now I'm going to show you the biological examples that have this same situation. They're just using actual nucleotides and amino acids. So here's what I'm looking for. This is my goal. This is my double-stranded DNA, and I know that because it's got a T. Again, it's double-stranded. I've transcribed it into my messenger RNA, and it's been translated into 6 amino acids. This is the protein I want. Now, for a missense mutation, a substitution has occurred. As a result of a substitution occurring, 1 amino acid changed. So the first, second, and third amino acids are identical. The fourth one is different, but the fifth and sixth ones are identical. So it's still making a protein, but is it making the exact same protein? No. We call this missense. 1 amino acid has changed as a result of this substitution. I did want to do a quick highlight on this condition, and this is the only one that I'm going to highlight because we come back and talk about this in another chapter. But sickle cell anemia, also called sickle cell disease, is the result of a missense mutation. So when I said the impact, it really depends. It depends on what gene it is and that individual and how much that gene is expressed. Sickle cell anemia is a condition in which the red blood cells, which are supposed to be shaped as so, are exactly, in case you don't know, they're exactly the diameter a red blood cell is of your smallest blood vessel, which is called a capillary. And when your red blood cells go through your capillaries, they line up in single file order. They cannot go multiples because those vessels are so small. But if they are this perfect shape, there is no problem. It just shifts, and I'm trying to... It goes right through. It goes right through. Single file order goes right through, and oxygen is dropped off and carbon dioxide is picked up. So it's sifting through the gases is what I was trying to say, and I was saying shift. And anyways, the gas exchange takes place there if they're perfectly shaped. Now, if you have that missense mutation where 1 amino acid is wrong, instead of being that perfect circle shape, they are a crescent or a sickle shape, and they're just a little bit taller than they need to be. So guess what happens when they get to those capillaries? They can't get through. They get stuck. And what about all those behind it that are trying to get through? Traffic jam. Everything just starts to pile up. Individuals with sickle cell anemia bruise very easily. Individuals with sickle cell anemia, because this is designed to hold oxygen, when it's this shape, it can't hold as much oxygen. Guess how they feel? Very tired. Very tired. I just need to have a rest. I just need to sit down. We see that all the time. Individuals with sickle cell anemia, if they stay in the same position, their blood could pool in a certain area. And you know that tingling sensation you get when the circulation's been cut off to an area, like, for example, you've sat on the toilet for too long? They get that all the time when they sit in a position. Like, they start to lose circulation. No oxygen gets to that tissue because of the way they're sitting. So they have to roll over. And let's say that they sleep really hard one night. They have a bunch of tingling and stinging sensation on one side of their body. So they have what we call good days and bad days. Some days they can't even get out of bed because of that. Sickle cell anemia does have a range, very mild to very severe. And you and I are going to talk about this again because we're going to talk about homozygote and heterozygote, dominant and recessive. But if you have the mild version, your symptoms aren't so bad. If you have the severe version, then your symptoms are pretty bad, life is pretty rough for you, and you usually expire early on, like in your early 20s. However, if you have sickle cell anemia, you're immune to malaria. Can you tell me where this would be beneficial? If you lived in areas where malaria was rampant, like, nobody knows where malaria is rampant? Not in the United States. Don't be like Mesquite. Okay, third world countries, specifically Africa. Malaria is really rampant in Africa, and guess where we see a high incidence of sickle cell disease? Now, if I were in Africa and I knew I had limited access to health resources, whether it was because I financially could not afford it or because they were not available to me, and I myself did not have sickle cell disease, but I was in my reproductive years, and I wanted to make sure that my child was taken care of, and I understood natural selection, who do you think I might choose to reproduce with? Somebody who might be carrying that characteristic with sickle cell disease so that I could make sure that my kid would be immune to malaria. And because I don't have it, they would just be a heterozygote, which means that they would not have the severe form. They would have a mild form. It would be a 50-50 chance. But if I knew what I was doing, that would be my choice. So when we talk about sickle cell disease, and we'll continue to go into that, it's a small amino acid was changed because of one silly substitution. And it impacted not just one function, not just carrying oxygen, but many functions in that organism. Okay? So that's a missense mutation. One amino acid is wrong, and that's just an example of the impact that it has. Nonsense mutations. Here again, I have my DNA, my messenger RNA, my healthy protein. In nonsense, a substitution is made, but it substitutes into a stop codon. So instead of making all 6, I just have 4. So I've prematurely truncated or stopped the synthesis of my protein before it was finished. This sentence makes no sense. This sentence makes no sense. A silent mutation is one. Again, here's my normal healthy gene. The silent mutation takes place, but because our genetic code is degenerate or redundant, the amino acid did not change. So this mutation occurred, and there was no impact on the protein that was manufactured. Nope. It's not dangerous because nothing changed. Good question. The last and final version is the frame shift mutation. So again, here's my normal example. But frame shift is the result not of a substitution, but I'm adding a nucleotide or I'm removing one. In this example here, we have we've inserted a nucleotide or base pair, and it shifts everything from that point down. And every amino acid from the point of the mutation down has changed. So there's still 6 amino acids. Here are my 6. It's just that only the first 2 are correct, and then after the point of mutation, those last 4 are incorrect. It did make a protein, but it didn't make the protein that I needed. Questions here on frame shift?
So when that happens, what does the body, what happens to that protein?
It depends. It depends on what I use that protein for. So if this is something that I'm only using it for a short time, perhaps I can take some type of oral medication to make up for that. But if it's something that's really big and meaningful, and I'm going to be using a whole lot of, then its impact is going to be bigger. And it also depends on where that gene functions. Does it function in all of my cells or just in a certain type of cell? if that makes sense. So there we go with our answer. It depends. It depends. Okay? Some examples, real-world examples of mutations that have occurred. First of all, mutations are caused by mutagens. A mutagen is just a fancy word for anything that causes a mutation. Okay? This first one I'm going to come back to because this is an example of a chromosomal mutation. But thalidomide. Thalidomide was an anti-nausea medication that was prescribed to women who were pregnant and in the 50s when they had morning sickness. And they took this medication over and over again because they were experiencing morning sickness, and they took it while their babies were small because that's usually when you have morning sickness, the first 3 months. So it impacted the entire environment for that child, that developing child. Well, the mom felt better, of course, because morning sickness, she wasn't feeling the symptoms of it. But when her child was born, this thalidomide caused the long bone growth in the child to be stunted. And so instead of having normal limbs, they were born with flippers. Was that a big mutation or a small mutation? I feel like that's a big deal. Now here, let me tell you the issue. The issue was the mutation, the mutagen was exposed to the environment when the baby was small and only a few thousand cells. Now, would that say, did it happen to the mom? No, because all of her cells were already formed. So we changed the environment of what that cell was exposed to, and we changed its gene expression. And now I have a kid with flippers. And I'm thinking to myself, I should have just thrown up for 3 months. Okay? But of course they didn't know, because if they did, I guarantee some parents wouldn't have done that. I hope most parents. Another example of a mutagen is radiation, like from the atomic bombs. Anytime an A bomb is detonated, the radiation from that spans not just in that area, but for miles. And it doesn't just disappear after a couple days. It's there for a long time. Now, if it's you and I in our adult lives and we're exposed to that radiation, it's going to cause some mutations potentially, which could result in cancer. But is it going to cause huge deformities in us? And the answer is no, it's not. Okay, it could cause cancers and tumors. Yes, there's no doubt about that. But if I were a developing baby that was really small inside my mom, only a few thousand cells, and I was exposed to that radiation over and over again, and I'm mutating at that level, it's not just in a few cells. By the time I'm born, it's in all of my cells. All of my cells. So the impact of when the mutagen is exposed, or when that individual is exposed to the mutagen, the overall impact is determined by how long and when in your life were you exposed to it. Okay? And of course, the radiation causes all kinds of birth defects. There's not just one type of birth defect. We're missing limbs, organs, miscarriages, lots of miscarriages. Okay, this first one is a chromosomal mutation, big deal. And I told you the difference with a chromosome is that it has multiple genes. Chromosome has multiple genes. So we have chromosome number 3, which should pair with chromosome number 3, right? Because they're homologous chromosomes. But in this case, chromosome number 3 paired with chromosome number 7. And then they crossed over. That should have never happened. But it's a random mutation. Nobody knew it happened. Anyways, as a result of this, in the cell that occurred in, it was used to fertilize a child. And so as that child was developing, the fact that crossing over occurred in those 2 chromosomes caused that child to be born without a brain stem. Can you tell me what's going to happen to this child? They're going to expire. They're going to expire. And if not without a brain stem, the brain developing outside of the skull, we still have a huge issue. We have a huge issue. So just small, random mutations that have large, very large impacts. And again, the timing is everything. for your final exam, not for your final exam, but for your exam coming up, you're going to need to know how to go from DNA to RNA to protein, or to amino acid sequence, or to polypeptide. Okay? You're going to need to know how to do that. Remember, I'm giving you the codon chart, but I am not indicating to you what the start and stop codons are. You are responsible for knowing those. Okay? Yes, ma'am.
Sorry,
No, go ahead.
The last one. So when a child is born with organs outside their body, is that the same thing?
So that we're starting to see that a lot more, and there's a lot of different reasons for that. And it really depends on which organs are outside of that child's body. Because now where we are science-wise, now we would probably handle this situation just a little bit differently. But now it's I don't want to say it's commonplace, but we start to see children developing more and more their organs outside of their abdominal pelvic cavity. So what we do is we wrap them in saran wrap kind of type stuff and slowly reintroduce them into the abdominal pelvic cavity. Now, are there complications for that child and their digestion and their defecation and all? Yes. But we have doctors who specialize in that, and we help to make that. Now, it doesn't necessarily mean, it could have been a gene mutation, or it could have been that when something was closing, it just didn't have the ability to close. Like, there's many different reasons for it. I don't want to say that it's because of a genetic mutation, because that is not completely correct.
So for the brain,
We know that because those two chromosomes have crossed over, that's why that occurred. Yes, ma'am.
Is it only those two, or can it be like
So,
3 and 6?
no. For those two, you will get that result. Does that make sense? So for those two crossing over, you will get that result. And that's happened enough that they can show that. Good question. Good question.
Question. Actually,
Go ahead.
I was born with mutations out.
Mm-hmm.
So why did that?
That's what I was just kind of trying to say. Like, there's no, it doesn't necessarily have to be a genetic mutation. I think that's what maybe right now you're trying to say, like something may be wrong with my DNA, but that's not necessarily the case. It could have been something within the environment while you were developing. It could have just been that as one process was finishing, like when we start to enclose everything, that there was just a small piece out, and that because it was out, it just stayed excluded. But that we know how to handle that, and we see that happening more and more. So we just reintroduce those intestines slowly into the abdominal pelvic cavity. And while that's occurring, we'd go through types of therapy like diet therapy so that the child can defecate and do all of that. And then normally they can function perfect outside of that once we fix that problem. Does that make sense? So this is the end of this chapter.